individuals, and coming on to ideas about tolerance or immunopathology, and how those immune responses are actually functional. How some particular immune response that we measure might actually end up in a measure of how well that immune response is able to be protective against an infection, provide resistance. And also believe, many of us are probably very much interested in ideas of measuring fitness. So this is, this is one of these slides that you just steal out of the first year immunology book. This is the idea that, as has been said before, particularly Steve's talk yesterday and so forth, that when we're talking about immune responses, we're talking about immune responses in the plural. So, over the left-hand side of the is the invertebrate immune responses, toll pathways, IND pathways, and other pathways that one can add on to that. This is very much stylized version of the vertebrate immune responses, T cells and antibody responses, but talk to any immunologist and they'll add in umpteen different varieties, flavors of T cells, eosinophils, natural killer cells, macrophages, a whole soup of cytokines and so forth. So, and certainly from looking at some of the posters yesterday, the issue is how to define what some of these useful measures might be when you go into a population of measuring cytokines or measuring cell response and so forth. So to get something which is functional in terms of something that we're interested in, and that might well be an ability to resist a particular kind of pathogen, or it might be some sort of more generic measure of immune health if one's able to define that in any sense at all. So this is kind of sort of going into the next few slides of some of my own work along with Mike Began and who's an oncologist, and John Bradley, who's an immunologist, and done largely by PhD students of mine, Andrew Turner. So it's done on, so this we see here is a field goal. So some of you might have read a science paper by Sandra Telfer on this system. So this, it's, as Vanessa says, it's a nice wild model. It's infected with a whole bunch of different pathogens, as Sandra Telfer's paper showed that those pathogens are able to interact. It's out there in the wild and it's exposed to environmental stress. It's genetically diverse, and in that sense, it can perhaps tell us things that rat mice can't do because it's got that kind of variation, both genetic and environmental. And at the same time, it's not like humans. No one really cares whether you cut out their spleens or not. So you can do all of those sort of immunological measures on them. So these things live in these clear cuts. So when the foresters are going around, they cut a bunch of wood and they sort of leave this sort of scrubby area where these voles will like, and they'll stand a good chance of going back at once time and catching the same animals if you want to do a longitudinal study. And similarly, you can also go in and you can destructively sample these individuals if you want to sort of do the kind of some of the immune measures that you might want to do that you would do with a rat mouse, take out its spleen, disassociate those cells, and either take unstimulated responses or kind of resting responses for a bunch of either cytokines or cell markers. So these assays are all done by real-time PCR in this case. The first thing we tried to do was to try to use ELISAs on the field bowls from wild mice, but those ELISAs just didn't work, and I think that's a problem that I think a lot of people have had in getting those cross-species reactions to work. So all of this is based on real-time PCR assays. Or one can take those spleen cells and stimulate them with various mitogens or antigens to try to sort of stimulate the kind of response that they might get if they were exposed to a bacterial pathogen, and see how much of a cytokine they're able to produce and so forth. And so these were, it's a reasonably kind of, so some of these cytokines were selected on the basis of the sort of doing different parts of the immune response, either inflammatory or anti-inflammatory, or Th1, but there are some missing, so it's quite hard to get a good measure of Th2 in here, for example, and that's just a feature of the fact that we're having to design primers at this stage and go from mice and rats to try to pick out these orthologs in field blooms. So essentially, what we're trying to do in this study really was, from my perspective, was to try to see whether we could find genetic diversity in these field voles. And did that genetic diversity, particularly in a bunch of immune genes, did that genetic diversity affect the types or the strength of immune responses that were produced from these measures? And in turn as well, because we've got a bunch of parasitological measures, because these voles are affected with a whole bunch of different things, could we also find that genetic diversity or genetic diversity of particular genes was associated with resistance to some of these different pathogens here? So trying to get at this individual variation. Then sort of go through the sort of GLM or GLMM type thing to try to control for as much variation as possible before going on and testing these genetic variation. So there are a bunch of, so on the left-hand side there, IL-1B, IL-2, IL-12B. So those are genes for which Andy's student was looking at genetic diversity, and there were also about 8 other genes as well, which he looked at, mostly cytokine genes, or other genes which have been associated with resistance in lab or human studies. And he was looking at genetic diversity in those genes using SNP markers to see whether there were associations, whether the genetic diversity of those genes there affected other of these immune parameters. And of the 8 or 9 genes that he looked at, there were these 3 here which seem to repeatedly come out with these significant associations with various immunological parameters. And those were basically either in an additive model or as asking, if you're a heterozygote, does that affect the value of the parameter that comes out? And finding these, so here, for example, you've got variation of IL-1, genetic variation of IL-1B affects the amount of IL-1B cytokine expression in these splenocytes, not surprisingly, but also this GATA3. And similarly here, you've got other associated genetic diversity. IL-2 affects IL-10. So all of these cytokines are linked together in a network. So one can imagine that some variation at one gene might affect the production of all the cytokines within the network, not just itself. So these kind of measures were quite strong and repeatable. And actually, I guess the kind of surprise for me is that when we looked at the size of those effects, and you compared the size of the effects that you found for these genetic variants, those were actually about as big as you would find for things like sex, or as well as we could determine things like the age of the animals, other intrinsic factors. So just the haplotypes that some of these genes seem to have as big an effect as other things that are much more obvious, like sex. Sorry.
So are the expression data for IL-1, is that a before and after design with the splenocyte stimulated in any way, or is that just what was in the tissue?
So that I think was stimulated one, yeah. So there was a little bit of kind of statistical reduction in here that sort of trying to take, if you had the unstimulated and the stimulated responses were both correlated, you could have took that correlated response to try to sort of reduce the number of factors that were done. But yeah.
Sorry, just another clarifier. The polymorphisms, were those in coding regions or in promoters, or both?
Yeah, so I mean the way that these were found were by taking cDNA and then sequencing those, and then trying to sort of go a little bit further to get the genomic corresponding regions so we could design the assay. So most of these SNPs are in coding region, but that doesn't actually mean that the SNPs we find are actually the things that are the causal mutations of it. There may just be a linkage to equilibrium with that. So typically, we've got 3 or 4 of these SNPs, and they're all in LD with each other, and presumably with other promoter region SNPs as well.
Okay.
And actually, in this it could well be quite likely that a promoter region actually amount of cytokine to produce or its responsiveness or whatever. Yeah.
Are you in the process of doing a whole genome variation on these bulls?
Yeah, so that's the grant I've just got to sort of get one of the slides to next on. Yeah.
I'm surprised that how much variation do you find among individuals? I guess I would think these genes would be really conserved. Do you have to look at a ton of individuals to get variation?
Yeah, certainly when Andy was doing it, so some of the genes you can find any variation, at least in the regions he was looking at, but most of them do. And similarly, so in the kind of molecular ecologists perhaps are a little bit behind certainly the human people and the livestock people. So the human studies have been doing this for a long time, looking at infectious disease, but also pathogenic diseases as well. Finding lots of variation there. So in one kind of general idea might be that you've got a particular cytokine variant that makes you good at resisting a pathogen, but makes you more likely to get kind of self-inflammatory or kind of some immune pathology. That's an idea at least, yeah. So the next thing after sort of saying that so this genetic diversity is associated with variation in these immune responses, again this particulization in genes, and test whether variation of those genes was associated with resistance to different kinds of pathogens. So we had these 2 data sets and a whole bunch of ticks and microparasites and so forth that we could measure for each of these. And so without looking in too much detail at this great unwieldy table, so the previous table I showed had IL-1B, IL-2, IL-12B variation of those being associated strongly with differences in immune parameters. And again, it's those 3 genes, diversity of those 3 genes, which seems to be dominating this table. That seems to again be sort of playing out in resistance to some of these different pathogens that these bulls have got. And again, just as previously, when you look at the sort of coefficients that are associated with variation at some of these genes, those are as big as things like sex and so forth as well. So that's kind of sort of where we're at. So the next slide, I think, is just a kind of summary of that. So this is essentially these 3 genes here: the IL-1B, IL-2, and IL-12B. Genetic variation at these genes being associated with sort of blue lines here going outwards are to the different immune parameters that we measured these real-time PCR assays, and the red lines going out here are to associations with these different parasite burdens. And what we're trying to do at the moment is to extend, so we've got more data into the next stage, possibly with that data, is to try to complete the thing a little bit more, to look at the variation of the immune parameters through season and how that affects some of these parasite resistances.
So just to clarify, resistance is being defined just by burden observed in the field and not by challenge assays?
Yeah, just by burden in the field, yeah. So this is primarily an observational study at least, yeah.
And this isn't saturating for those 3 genes, right? These are just the ones.
Yeah, so there's a rather arbitrary selection of genes that my PhD student had time to analyse, and so you started before all the next gen sequencing came out. So yeah, I mean there will be more, so we've not done all the cytokines yet. So these were sort of 3 out of 8 or 9, which were picked on a sensible basis. But there were others there that tried to amplify but couldn't, like IL-4 and so forth, and other cytokines.
I mean, there are always genes that have been picked out for instance, sensitivity to diabetes.
Mm-hmm.
And I think there are, 19 loci that seem to confer differential effects in it. Not only diabetes, but infectious diseases. How come I don't see any of them right there?
Yeah. I mean, we didn't go for the MHC.
I thought it was a big.
Yeah. I mean, so the reason we're not doing that is the molecular ecologists have been going after the MHC ad nauseum, so I just kind of wanted to sort of push it away to the rest of the genome from that. Yeah. But yeah, you're absolutely right. So we're extending it out on a more sort of systematic basis is what we do with the ad. The whole genome sequence.
It's a great system. I mean, once you use next generation sequencing to do the genetics and then the susceptibility website.
Yeah. Which brings me neatly on to my next slide. Yeah. Just going to run with Mike Began and with Amy is in the audience, and that is doing this genome sequencing. So at Liverpool and at other places, there are these next generation sequencing machines, and it's now pretty much practical to do that kind of genome sequencing for non-model. So what we wrote in the grant was to do a transcriptome and then to pick out get those coding regions and use that to pick out stuff by sequence capture. So this technique where you use oligo arrays to fish out corresponding genomic regions and sequence what sticks to those arrays. But I think actually what we'll probably do is to do a stage further, which is just to stick field ball here through the HiSeq, give it a few lines of HiSeq, and then compare that either to the mouse or rat genomes, but the Broad Institute is also doing, I think, the prairie vole, and then at least that sort of fairly low coverage of sequencing, given something relatively close to it, we ought to be able to pick out at least the coding regions and the genic regions from that, and hopefully get as close to a complete suite of genes as we can, particularly immune genes and also control genes from that. So that the kinds of things we want to do with that are to look at particularly genetic diversity within populations, which are the genes that are most diverse, because that sort of points your eye to things that, particularly immune genes, that might be co-evolving with the pathogens, or for genetic diversity is most important, particularly if you can run the sort of population genetic tests that we like to do on that, looking for balancing selection or selective sweeps, that kind of thing. But also Amy has been working on the wood mouse. And apologies, Amy, if you've already used that gag elsewhere. So she's been doing kind of a more experimental approach and has been saving samples from that. And so again, the wood mouse is sort of relatively close to house mice. So again, we want to generate at least a basic genome sequence for that and use that as a basis to both to be able to look at diversity within genes, but also it's got knock-on effects that once you've got the basic gene sequences, you can go off and you can design real-time PCR assays for whatever it is you want to do. Get a more complete, exhaustive set of immune assays from that. And also, if you've got comparative species, there's actually in the population genetic techniques, that's much more powerful than being able to look at those genes which have been perhaps under selection, which have been driven to be more divergent between species. And if you've got as well a measure of diversity within species, that acts as a nice control, and there's lots of ways in which you can try to work out which genes are being driven by selection, presumably being driven by those host-pathogen interactions. That points you right to things that might be particularly important, and then again one can go out and look either at individual SNPs in those genes and compare that to resistance in a population of, I think it would be probably about 1,000 animals for each of the field voles and the wood mice there. So, consider more kind of discussed before. Yeah. So, also just got a graph with Mike Boots. So Mike was talking yesterday about this Podia system and about how the evolution of resistance was dependent on the amounts of resources that you have. And so again, it might be the case that those mechanisms for resistance change under different resources, and we can test this if we look at microarrays and gene expression on the podia. And actually, the way in which we'll probably do this is just to get a genome sequence for the podia first of all, and then once you've got the complete set of genes, you can then create microarrays which have got the entire suite of genes on it, and then you can try to work out how it's responding to viruses or whatever, how that changes under different resources, or you can do look at the outcome of selection experiments and look to see what genes have been responding to selection. So it's all sort of quite exciting at the moment in terms of the ability to do this. I mean, even in between sort of putting a grant in and getting the grant awarded and then actually getting the grant started, and the technology moves on so you can actually do a lot more than you intended to do. And I think actually there's going to be no particular barrier for working on non-model species, whether that's birds or whether that's other rodents. It becomes easier and easier to get genome sequences for those individuals. That the cost that one can easily justify on a standard grant proposal. And much of this, of course, is driven by the sort of new methods in sequencing, and driven particularly by the medics. So, I mean, the goal certainly was to get a $1,000 genome, which we'll probably get at within the next year or 18 months or so, and then after that, down into $100 genome for individual humans. But actually, the technology that one uses for that, one can just as easily use for non-model species as well. So, if you could sequence your non-model birds or rodents out in the wild for $100 each, then one can imagine doing a study design where you just take 1,000 individuals and you sequence all of those individuals. So that might be the next sort of population genetic study that you have, getting 1,000 genome projects within perhaps the space of a postdoc or a PhD student grant. So previously, those kinds of 1,000 genome projects, population genomic projects, have been within the remit of the sort of big collaborative centres. But now I think that's going to be much more democratic and able to be done by individual labs. So these are just so some of you probably get this kind of sexy technology talks all the time, but so just kind of some of the technologies which are sort of on the horizon for this. So this was early in the year. This was announced. This sequencing system here, so it's a single molecule sequencing system done by Oxford Nanopore, and it kind of it's as far as I understand the sequencing technology, you've got a little sort of a pore in a membrane, and you sort of squeeze the DNA through that like toothpaste in a tube, and as it does it by some biochemical magic, it reads off the sequence as it goes through that pore. And that kind of single molecule sequencing, if it gets cheap, has got a number of advantages. So one advantage is that you get quite long reads from that, 50 kb reads or so. And another advantage is that you don't need very much template at all. It's just single molecule. And it should, one hopes, be cheap because of the single molecule. It's not actually taking up that much by way of reagents. So you can either get these kind of sequencers here, which essentially you can just slot in what's like a video cassette into it with your DNA in it. Or there's here, which some of you'll have seen, which is like a USB stick, and that's actually the sequencer on there. And you can stick that into the side of your Mac, along with the blood sample. That takes fairly minimal preparation, and that will produce about a gigabase of sequence in this iteration. And doubtless that will come on, and you'll end up being able to do much more sequence from that. So that makes it able that you can do this kind of sequencing in your own lab without having to chat up someone from the Broad or someone from BGI or whatever. But also, you've got these little USB sticks. You can take those out into the field. You could do, metagenomic studies. You can see what kind of bacteria and cells are in the environment from that kind of study. At the moment, these cost about, I think it's about $900 or so that they're going to be selling for, which is maybe a bit much at the moment. But as I say, that cost will come down. So this is another competing technology, and one of the good things is that there are several of these technologies coming through, and they're all competing against each other, and that just sort of helps them to sort of raise their game. So this is like a little silicon chip here, and so their advantage with this is that they can, instead of having their own bespoke plant, they can actually just kind of contract it out to the microchip factories in South Korea and so on, to produce these. And essentially, so when you add a nucleotide to DNA when it's extending, that releases a hydrogen ion as that nucleotide incorporates. And so there's thousands of little wells in there that essentially act as little pH meters. So each time hydrogen ion is released, it changes the conductance of the silicon chip. And that's read out by one of these machines here. So each of these machines here for the ion protein, this is supposed to be about $150,000, but one of these chips here is going to be perhaps sort of $300, $400, $500, something like that. So sort of quite cheap, and probably able to do tens of gigabases. Certainly the ones are probably going up to 100 gigabases or something like that. So that's the sort of technology with that. So I guess just to kind of give one or two other potential sequencing applications. So I think this is actually quite an interesting sequencing application that I've not seen being applied to a wild system yet. So this is sequencing the T-cell repertoire. So as we go through life and we get more pathogens, our T-cells rearrange, as you know, so those T-cell receptors in the DNA, or when they're expressed into the RNA, you can sequence those, and you can get a flavor of how diverse an individual's T-cell repertoire is, or to see what kinds of particular kinds of T-cell receptors an individual's got, and then does that repertoire change between different individuals that they've been exposed to different pathogens, or according to their early life history of pathogen exposure. So it might be possible to get some kind of readout of what the T cells have experienced and what the repertoire is, which I think would be a really neat thing to do in a wild population. And on the right hand side here, so this is something which is the metagenomic sequencing, going into an individual's gut and doing, typically it's 16S sequencing, and just counting the number of different kinds of species, the different kind of abundances which come out from those, and those metagenomes have a big impact, the biota, on how the immune response is directed. Some people are trying to link these T-cell receptor sequencing with the metagenomics and so forth. And as the sequencing technology comes forward, particularly with these long reads, instead of just being able to do 16S sequencing, there's a lot of interest in just being able to do whole genome sequencing from this kind of mixed soup of bacterial DNA that's extracted, reassembling those. And particularly, I think, when one's thinking about horizontal gene transfer, so, two bacterial clones from the same species might have very different complements of genes, because bacteria swap the genes around quite readily, and so that's getting those whole genome sequences, I think, would be quite exciting. Thanks for your questions so far on that.
Can you do this receptor sequencing on single cells?
So the question was, can you do this T-cell receptor sequencing on single cells? Yeah, there's no reason why you can't. It would just be a Sanger sequencing approach, really, because one cell will presumably just have one T-cell receptor.
Would you like to associate the alpha and the beta sequence?
It's true.
Do that in computers.
Yeah, that's right. So this kind of T-cell receptor sequencing that's done by NextGen is sort of a soup of, yeah, exactly, yeah. So this is the kind of omics word boggle. This is genomics is the next big thing when you go to these bioinformatic or genomic conferences. So I've just told you that sequencing genomes is going to be completely easy, and aside from the bioinformatic challenge of that, the next trick will be to actually associate that genetic data with useful phenotypic data, because actually most of the questions I think that people will be talking about are not really about the sort of genetics that I'm particularly interested in. It's about the phenotype of the organisms and so forth. And that's a more challenging thing, obviously, because once you get a genome sequence, that's what the sequence is, give or take. But the phenotype space, all of the potential number of stuff that you could measure about an individual is completely unlimited. So some of the things that one can measure from that, still sort of lie within the kind of omic sphere. The transcriptome of a particular bunch of cells or a tissue, or the metabolomics, and there's a poster on that. These metabolites circulating around in the blood, or trying to work out what's going on in the gut. The other hard technical challenge, as far as I can see, and I'd be interested on getting some opinion from card-carrying immunologists here, is so the kind of studies which I've done with the Vol system was all about these real-time PCR assays to try to get immune measures from that. But actually, potentially more useful is cell-based assays or ELISA-based assays based on the protein circulating around the gut, and that's a much harder thing to do for non-model species. So we weren't able to get those ELISAs raised in mice to work on the balls, and I think one of the reasons for that perhaps is that when you generate an anti-IL-4 antibody against mice, you do that perhaps in something like a hamster, and so what the hamster is recognizing is stuff that's unique to mice, and the stuff that's perhaps common between mice and hamster doesn't generate a good antibody in the hamster, and so actually what you end up with is not the stuff that's conserved in those antibodies, it's the stuff that's different, and so it seems a much harder slog generating specific ELISAs for each non-model system that individuals are interested in, which is why we went down that real-time PCR approach. But you do lose some information from that.
So none of the different antibodies for cytokines cross-react? The reason I'm questioning that is that oftentimes they'll raise antibodies in mice, monoclonals, and nonetheless they'll still respond to mice because you broke a tolerance somehow, and you're looking at intracellular cytokines or intracellular molecules that aren't necessarily tolerizing the immune system. It just didn't work, though.
Yeah, it was, certainly our observation here just didn't work. I mean, some of the ones from so Jan, who did all of this, Jan Bradley. So she had tried some wood mice samples, and so some of those would work, I think. But again, it's quite a limited set. You have to sort of go through quite a lot. It's quite expensive and quite demoralising. Full stop. Yeah.
I suppose what you'll do soon would be to stimulate the cells and then just do a transcriptome in RNA-seq, and then just look out the frequency of cytokine messages to all this.
Yeah, and sort of scaling that up again onto lots of samples, I think, is expensive. Yeah, because the library prep side is still going to be expensive,
Right.
but. I mean, I guess if you're doing these kind of bioplex assays or whatever on cytokines, that's sort of $6,500 or something. Each assay, if you've got 20 cytokines you're looking at, so maybe it's not that much difference from that. But yeah, I mean, I'm not an immunologist. I think the trick is to get the right cells and the right kind of stimulation that you're doing that that proper immunological assay.
Yeah, I don't think that'd be too difficult for any of the immunologists around.
I mean, perhaps. Well, perhaps one of the harder things is to interest the immunologists that this is a useful thing for them to be doing, and it's nice that you're here and some of the other immunologists. But yeah, a lot of the immunologists just are much more interested in the mechanistic side of things. Yeah. So they're sort of trying to. I mean, this was kind of raised at the last meeting, from what I remember, is trying to sort of raise the profile of some of these non-model wild systems and sort of show what one can get out of that. Yeah.
And one of the things that we really want to do is some sort of non-invasive gene expression assays. So are you guys doing expression from blood?
Yeah, so we're doing that at the moment. Yeah, we've not analysed those data, and we don't know how well that correlates with something that's functionally informative in the spleen so far. We would hope that that it would be informative of that. I mean, so for that, we'll all we'll be able to do is take blood and put it into RNA later. So the only thing we can measure are the unstimulated responses. Whereas in some human studies, they've got the ability to take a larger volume of blood and then stimulate those. Yeah. So I guess some of the other things that we might want to do in this kind of genomic space is to try to work out how to do behavioural assays in a kind of automated way. So this is something that caught my eye in Nature Methods just a couple of months ago. So so you might be able to see sort of little lines around these mice. So this is all sort of done by image capture, video capture, and sort of just recording the behavior between these pairs of mice, and they're using this to try to find genes involved in changing these behavioral responses. So I think it would be nice if one could think of ways of getting behavior automatically recorded in a wild or a semi-natural system. So this is kind of video capture way of doing it, but you can also possibly sort of give little tags under the skin, and each time they go through a particular gate, that trips that off. What are the the individuals that are most curious are those the ones that are most exposed. So somebody had a bold shy fish poster out there, so it's a similar kind of idea to that. But I think being able to just sort of scale up these phenotypic assays in the same way that I've been talking about scaling up the genetic assays, I think that's a challenge. I don't really know how to go about that, but it probably involves interaction with engineers and trying to come up with clever solutions. But I'm sure there's I'm sure there's nature papers out there for people that can come up with those clever solutions and apply them to interesting questions. Yeah, and similarly, so implantable devices. So you can buy these devices to monitor your lab mice in terms of blood pressure and that people are interested in. If one could do that simply in a wild population or a semi-wild population, having that sort of continuous monitoring of some internal physiological state as well, I think that would be really, really an exciting thing to do. So this comes from this review here, and this is having put this up, I've still not quite got my head around the implications of it. So this is one way to think about a genotype phenotype map here, and perhaps to try to identify pinch points within this map. So the kind of example that's given in this review here is if you're interested in looking at diabetes, then actually one of the most interesting states that you can look at is a kind of internal phenotypic state, which is obesity. If you're obese, you're much more likely to be to get diabetes out at the other end. And so actually one could do all of this sort of work on genetic diversity, genotyping, but actually much of what you're measuring is perhaps the propensity of an individual to become obese, and that's going to be dependent upon the environment that an individual is going to be in. So actually measuring obesity, if one is sort of interested in a biomarker for that particular phenotype, that might be the thing to go for. And I've not really got my head around this properly, but when people talk about competence or about health or about those sort of concepts, which are perhaps a little bit vague, but we kind of know what we're talking about with that. Can we sort of formalize that in this kind of way to sort of identify some set of measures, some parameters that sort of correspond to those pinch points. So that's maybe kind of thing that might come up in some discussion later. So last slide, I think this is just thinking the kind of challenges that are generated by this, and the thing that is skimmed over really is the bioinformatics. I mean, so it might be easy to generate 1,000 genomes, but analysing that is always going to be a complete bugger to do. And so in our lab we've got this server system with 400 cores of processors and huge amounts of hard drive, and we're just spending tons of money adding new hard drives, adding new cores to this, and the nature of what my postdocs do is changing radically. So it used to be that when Andy did the Ball study, he would spend his entire time in the lab with a set of Gilsons in front of him. These days, so he's coming back into my lab to do a postdoc, and the nature of his job is going to change entirely, and a lot of what he's going to be doing is handing a DNA sample to our genome sequencing center. They're going to stick it through one of these fancy machines, and then he's going to spend the next 6 months, a year, analysing that data on a computer, and that involves getting a lot of skills into that postdoc community, into PhD students, and getting people with the appetite to do that, and then linking that sort of genetic data to to those immune measures, to the resistance of individuals and so forth, raises quite substantial statistical challenges for that.
If you're just like a disease ecologist and you want to do some of this stuff, are there bioinformaticians out there that would be interested in collaborating? How would you recommend starting this? Obviously, in my lab, they're not going to skills to do that.
Yeah, I mean, if we talk about what we do at my sort of sensible genomic research. So we've got people coming to us and sort of studies and sort of ecology type things that we kind of offer with various sort of kinds of services. So sometimes they'll have a biocompetition in our lab for 6 months, 12 months, something like that, painful, or whatever the grant is funding that. And that's actually kind of quite a nice way of doing it, that you've got someone who's kind of paid at your disposal, but who's got those skills. And it still needs the PI's knowledge of the biology of the system as to what questions to go after on that. I mean, another way of doing it is to work with a card carrying biocompetition who knows about machine learning and all of those kind of things, or a computer scientist. But typically they might be interested much more in what's a sexy algorithm rather than what's a sexy exactly. So it's trying to sort of get that balance right between having whoever it is that's doing it actually interested in both the computational side and in the biology of that. I think long term, I think that's going to come from getting grad students skilled up and tooled up in those kinds of methods, because that's the nature of what a lot of them are going to be doing. Yeah. But hopefully, I mean, it might be the case that as more and more people are doing a similar kind of thing, that more programs become available that just make that job easier. But there's still going to be the requirement to do that.
What's your sense right now in terms of the number of bioinformaticians who are out there? I mean, as an example, there was a job search in Canada for one, and the university eventually closed the job search because they couldn't find any qualified people. And I'm wondering if the technology is changing at a faster rate than the people who are able to actually understand the technology.
Absolutely. Yeah. So similarly, we had a job ad out for a head bioinformatician, and it was only the third attempt, and with a lot of persuasion and incentives that we could actually get someone good to do that job. Yeah, and it is really difficult to find those people.
Also, if you're going to hire an academic bioinformatician, then they want to do fancy algorithms.
Yeah.
And that's what they're going to be judged on, whether they can publish in those journals, not whether they can help you do your
That's
research.
right, yeah. I think, and that's absolutely true. I think the only thing that perhaps the sort of ecology, disease people have got going for them is that we tend to be quite numerous anyway. So it's perhaps easier for us to sort of get those skills within our grad students or whatever that it would be. Many of the sort of cancer guys just know about doing PCRs or particular genes, and they've shied away from that whole numerical side sort of right throughout their career. So I don't really have that much else to say, and I guess it's just about thinking yesterday about whether something like immunocompetence is actually a useful concept. About is there given that there are this kind of multiplicity of different immune responses, is it a single thing that we can define as immunocompetence, or do we sort of just measure a whole suite of different genotypes, phenotypes, and then try to post hoc recognise what's important in terms of those pinch points that I showed in the previous graph. So I think those are sort of challenges in terms of trying to interpret the data, and also in terms of trying to design those kinds of studies. So that's as much as I've got to say, and but hopefully these are the time for discussion. Sure.